Thank you, Dr. Molisco, for the introduction and the opportunity to speak today. Also, Dr. Daris and Dr. DeGroot. Really excited to share our unpublished work looking at how mutation status and single cell sequencing might affect our understanding and potentially inform clinical decision-making for patients with melanoma brain metastases. My day job is as a radiation oncologist, specializing in CNS malignancies of all sorts, and pretty much all of the other parts of my life outside of that day job is as a physician scientist. And really, I'm a new faculty member here in radiation oncology and neurological surgery. And my lab focuses on understanding how growth factor signaling through receptor tyrosine kinases and the RAS signaling pathway is important in nervous system tumors, with a particular emphasis on NF1. And so perhaps given all of those biologic interests, it's not surprising that melanoma brain metastases caught my eye. This is a context where the RAS pathway is critically important. Genes such as NF1 and RAS, RAF are commonly mutated and in fact are a big part of how we think about therapy with drugs like dabrafenib and trametinib. The work I'll share today will really maybe speak to my scientific training as a geneticist and some of my technical expertise in bulk and single cell genomics to understand how different melanoma molecular groups might behave. Let's see if we can get this working here. Perfect. So starting with disclosures, no disclosures to report. And so maybe taking a big picture point of view in terms of how historically we have thought about the management of brain metastases. I think in the last 10 years, we've really seen a big revolution with the advent of CNS penetrant agents. And so when I started residency, in addition to the clinical status and clinical component of every patient we saw, this is really how, from a imaging and decision-making perspective, we thought about brain mets management, which is this was a disease and entity that we needed to treat with local therapy. And so the two axes that we're taught to think about this are number of lesions here on the x-axis. You saw Dr. Bronstein's talk reference the Japanese study where 10 brain metastases was a magic number of which we could SRS. And then you saw Javier, Dr. Villanueva talk about up to 60 being our record here. So clearly that x-axis is subject to change. The other thing we think about is the size of the lesion. And again, in historic radiation surgery trials, 3 centimeters was about the limit of what we could radiate safely. And so maybe just to walk through this very briefly, for a low number of large lesions, often these are symptomatic. And so we want decompression surgery is often the way we go. For a low number, up to 10, perhaps more, of small lesions, SRS, stereotactic radiosurgery is often our preferred approach. Historically, we have done a lot of whole brain radiation therapy, WBRT. This is not now really reserved for patients who we think have a limited life expectancy and are not going to suffer some of the neurocognitive side effects associated with whole brain. And then the other thing to mention is that BSC, best supportive care, increasingly something we'll employ, particularly for those poor prognosis patients. How does this change now that we have all these systemic agents that get into the brain? One of the ways I think about this is now we can often, as we've heard, for asymptomatic brain metastases, perhaps start with CNS penetrant agents and certainly combine them with surgery, radiation, in some sort of sequence. And perhaps one way to conceptualize this is if you start in this intermediate ground, getting systemic therapy can perhaps push you into a good direction, but also potentially into a bad direction if we lose our window to treat. And so I think it's important to think about when and how we use these agents. Now, I think Dr. Molisco's made this excellent point multiple times today that brain mets are not one big bucket. There are probably many different buckets. And so really to focus the conversation today and for our research work, we're focusing on melanoma brain metastases and how we can incorporate molecular information in this context to help guide some of these agents. So given my interest and background in the RAS pathway, I could not give up the opportunity to talk about the signaling cascade in front of this audience. And so really mutations in the RAS pathway are essential to how we define molecular melanoma subgroups. And this also informs CNS penetrant therapy. In its most basic form, the RAS signaling cascade involves the RAS protein, the most commonly mutated oncogene in human cancers, which can activate RAF, which can then activate MEK. NF1 is a tumor suppressor that serves to turn off RAS. And in fact, these 3 genes, NF1, RAS, and RAF, are the basis for the TCGA molecular classification of melanoma. which I've helpfully put up here. Essentially, what you can see here is that the three main commonly mutated genes that make up these subgroups are BRAF mutation, RAS mutation, NF1 loss, and then this kind of mysteriously named triple wild type group. Turns out if you look at this triple wild type group more closely, many of them have mutations in other RAS pathway components. So really, we think all of these subgroups are RAS-driven tumors. How does this inform therapy? As I alluded to earlier, and you heard a little bit about in Dr. Dowd's talk, we have agents such as dabrafenib, which targets the BRAF V600E isoform, which is a commonly variant found in these tumors. We also have trametinib, which is one of many MEK inhibitors. And what I would consider a landmark study in 2017 showed that dabrafenib and trametinib can actually be used in BRAF V600E mutant melanoma brain mets. And then certainly I cannot talk about melanoma and melanoma brain metastases without bringing up immunotherapy. And as you heard from Dr. Dowd, combination ipi-nivo is now something we do, particularly for asymptomatic brain metastases. Patient selection remains a challenge for who can get these agents up front, but definitely something that we think a lot about in the clinic. I think I finally figured out how to use this remote, so there will be less moving back and forth and less mistakes with the laser going forward. So in terms of what I'm going to talk about today, really three main avenues to discuss. First, I will talk about how our molecular analysis of resected melanoma brain metastases using UCSF 500 can start telling us how different molecular subgroups work. The bottom line here is we will show that we can recapitulate the 4 TCGA melanoma subgroups in brain mets, and there does seem to be a prognostic implication with those different groups. The second thing we'll talk a little bit about is single nuclear RNA sequencing of treatment-naive melanoma brain metastases. Two things to unpack there. You've heard a lot of beautiful work when you go about single cell sequencing. There's a little twist here with single nuclear sequencing, which involves frozen samples. And as everyone here can appreciate, getting fresh samples, especially in the COVID era, is a big challenge. And so being able to get high quality single nuclear resolution from frozen specimens is critical. And so we've come up with a pipeline to do that. And then I think in all the talks today, we've heard a lot about the different kinds of treatments patients get and how that can change the landscape of what we're looking at molecularly. And so we really focused on treatment naive metastases for that reason. And then a little bit about ongoing and future projects, because I think the point of today is to collaborate. It's kind of funny, having been here for 5 years now, there's still people I see all the time. I don't know what they're working on. So it's kind of fun to see all the talks this morning. So beginning with the description of the cohort in which we looked at UCSF 500, we actually have this data up now through a internal CBio portal instance. So anyone at UCSF can request access to this, and you can essentially pull mutations for patients that have had UCSF 500 profiling done. And then if you have an appropriate IRB, you can get the de-identified clinical information for those patients as well. So very powerful resource compared to the old days of where I would have to go through Apex and look at these scanned clinical documents to figure out what mutations folks had. Using this approach, we were able to identify a group of 60 patients who underwent resection of a histopathologically confirmed melanoma brain metastasis with paired UCSF 500 data. In the interest of an oral presentation format, I didn't put up all of the clinical parameters for these folks, but some of the most pertinent things in my mind. The median age is 62 years. The median follow-up is a little bit over a year and a half. This is a retrospective study, no controls on what patients got for treatment. And so in this cohort, there's a little bit of a dog's dinner of systemic therapy type. 85% got checkpoint inhibition at some point in their treatment course, whether that was IPI, Nivo, both, and 35% of them got dabrafenib and trametinib combined MEK inhibition. You'll notice those numbers together add up to more than 62, and that's because you can be in both groups if you got both sets of therapy. The other thing is timing of treatment, which from a clinical decision-making perspective is something we talk about a lot when we're managing these patients. 57% of them had treatment pre-surgery and 67% had treatment post-operatively. Again, many patients might get both pre and post-operative therapy, so it does not add up to the total number. And so the very first thing we wanted to do with this UCSF 500 data was look at the mutational landscape. What are the mutations that we're picking up in this cohort of melanoma brain metastases? You could imagine one of two scenarios. The first is brain metastases aren't that special molecularly compared to primary melanoma, and you get the same 4 TCGA subgroups. Or you could imagine there's something really special about the brain, and I'm a radiation oncologist focused on the brain, so I hope that would be true. But it turned out that was not true. We still do see the 4 classic TCGA subgroups. A third of them are NRAS mutant, a third of them are BRAF mutant, and a fifth of them are NF1 mutant. For the most part, those are mutually exclusive. There were exactly 2 patients that had a mutation in 2 of those different genes, and that's something to maybe dig into at a different time. Somewhat interestingly, the most common mutation we saw was actually not any of those, but it was a TERT mutation in the TERT promoter. So 3/4 of our patients had a TERT promoter mutation. Turns out that's been described in melanoma in the past, and as I'll show you in a little bit, I think this is probably not something that's driving the biology of these lesions, but something that certainly I didn't expect to see when I walked in. So simple things being simple, the very first thing we looked at was outcomes based on molecular subgroup. What I have here are Kaplan-Meier curves. X-axis here is time in months. The y-axis is percent survival. I have colored the patient curves by molecular subgroup. So in red, we have NRAS mutant, which is again about a little bit over a third of patients. In blue, we have the BRAF mutant patients. In green, we have the NF1 mutant patients. And in black, we have the triple wild type patients. And you can hopefully appreciate, even for the folks in the very back, that there is separation between these curves, and that the NRAS mutant and BRAF mutant subgroups have significantly worse outcomes in terms of survival. The BRAF association has been reported a bit in the literature. And as Dr. Dowd mentioned a little bit in his talk, this is something that has come up. I think the NRAS mutant prognosis is a little bit newer. NRAS, in my mind, is kind of the ugly stepchild of the NF1, BRAF, NRAS triumvirate here, in that we have MEK inhibitors and BRAF inhibitors, but we don't have great NRAS inhibitors, although those are, I think, increasingly coming down the pipeline in my day job as a physician scientist. The other thing that I promised I would bring back up again was this TERT promoter mutation that we saw. Turns out no prognostic significance. Black here is the TERT wild type patients, and red is the TERT mutant patients. And so something I think I've been asked every single time I've circulated an abstract or draft of this manuscript is, how does this mutation status relate to treatment? And the answer I've always given is it's very difficult because the treatment characteristics and timing are very heterogeneous in this population. But I figured this was an opportunity to put this data out there, at least with all the caveats that I cannot control what patients got or when they got it. On the left, I have the NRAS mutant subpopulation. On the right, I have the BRAF mutant subpopulation. I think what you can appreciate, at least for the NRAS mutant patients, timing of immune checkpoint inhibition, which all of these patients got, did not seem to make a big difference in their outcome. And if I look at the BRAF mutant patients, 16 of these folks got immune checkpoint, half preoperatively, half postoperatively. 4 got dabrafenib, trametinib rather than immune checkpoint, and that's that dashed line. And I think sample size is really just too small to say much about whether there's a difference here. One thing we're still interested in doing going forward is expanding this out. And perhaps as more patients are getting CNS penetrant agents, this will become something that we could think about more robustly. So switching gears a little bit and kind of moving, quote, from the bedside over to the bench in at least one capacity. I want to talk a little bit about our efforts to do single nuclear RNA sequencing of treatment-naive, fresh frozen melanoma brain metastases. For me, when I was selecting the patients, which is really a critical part of any single cell or genomic study, I really wanted to focus it on this question of BRAF mutation status and the fact that this does seem to consistently confer a worse prognosis. And so what we were able to identify was a set of 6 patients, one of whom came straight from the OR before COVID kind of shut down all of our ability to do that, and 5 of which came from the biorepository, which I know Joanna is going to speak about in a moment. So kind of shout out to Joanna for helping make that happen. In these patients, we also had a 1.5-year follow-up. Systemic therapy type, again, this was one of the big criteria I tried to put on which patients we got. All of these were treatment naive. So you can then imagine a lot of these patients were actually resected before the era of CNS penetrant agents. So these are actually older specimens in general. And then, like I mentioned, I kind of did this on purpose where I had exactly half of them be BRAF mutant to try to make a comparison compared to BRAF wild type. And so before I get into the biology, I think we've seen a lot of beautiful single cell and sequencing data today from Hugo and others. And I will at least talk a little bit about the technical aspects of the wet lab side, just because this is, again, a point of being here is to enable other folks to hopefully use these approaches in their own work and experience. What you start with is a frozen tumor specimen. This can come out of the OR, come out of a cryo vial, or come out of a tube that's been at liquid nitrogen -80 for God knows how long. And then you have to somehow get usable data and tissue out of that. We trialed and errored a lot of things. When I started doing this in 2017, there were really no published protocols. In the last 5 years, there's now probably a dozen or 2 dozen published protocols. We kind of learned the hard way and with those people how to do this. And the protocol we came up with, which I'm happy to share with folks, is we essentially mince up the tumors, down some mechanically, dissociate them with a combination of collagenase and disphase, which are enzymes folks commonly use, fix the nuclei so they don't break apart when we run them through our microns and our microfluidic devices, filter them, and then count them. And then at that point in time, we have now our DAPI stain nuclei. We can run them through a microfluidic device to do the single cell sequencing. I think in this scenario, a picture is worth 1,000 words. So what we have here on the left are the DAPI stained nuclei, and they are in a hemocytometer for us to enable cell counting. You can see there are not clumps, individual cells at a sufficient concentration to get usable data. And on the right, this is one of my favorite photos that I think I've used in all of my talks over the past 2 or 3 years, which is these are the nuclei actually encapsulated within microfluidic droplets. The whole basis for single cell biology is you can use droplet microfluidics, which are really, really tiny volumes of water, oil emulsions to put individual cells or individual nuclei in isolation, do your molecular biology in those droplets, and then barcode them or put a specific DNA sequence on them so you can deconvolve single cell data. And what I think you can see really beautifully here is there's, for the most part, almost all of these droplets have just one nuclei. You can see that I showed in red a couple of droplets which have 2 nuclei. So don't be fooled when people say single cell sequencing. It's really that 95% of them are single cell. The other 5% are multiplets, and you got to keep that in mind, especially when you're dealing with heterogeneous samples like these. That's the workflow, and again, happy to share that with folks. And essentially what we then have to do, this is similar to things like 10x genomics, which is now commercially available. Again, when I started, that wasn't something 10x could do. So we had to kind of homebrew this with our collaborators in the Abate lab. We encapsulate them, lyse them, barcode them, amplify the cDNA, and then make a nice sequencing library. And if you see this on a bioanalyzer, then you know your work is done. Usually there's a lot of blood, sweat, and tears that goes into generating this curve. And I was happy to report we were able to do this at least 6 times and hopefully more going forward. So you've seen a lot of Umap plots today already from Hugo, so I won't belabor some of the technology and computational approaches and how we do this, other than to say essentially what we get out of a single cell data set is some number of cells or nuclei. In my case, I got 35,527 nuclei. So these are individual cells that pass all of our quality filtering across our 6 melanoma brain met samples. Now you essentially have a matrix then where your x-axis is every individual cell and your y-axis every individual gene. So it's a giant matrix, 36,527 by 28,000 genes in the genome. So somehow you have to reduce that into something that humans like us can look at. The way we do that is with something called Umap. There are many other techniques available. Every single dot on this Umap plot is an individual cell. And the x-axis and y-axis here is a relative measurement of how similar cells are to each other in this space. So dots that are really close to each other are very similar to each other. Dots that are further away are less similar to each other. And then we will use often something called a clustering algorithm to color these dots to say how similar or different they are to each other. You'll notice that the coloring, sometimes there's one blob that has multiple cluster names on it. The reason that happens is I'm projecting this into 2 Umap dimensions, but we draw our clusters in 20 Umap dimensions. So probably the difference is in a different orthogonal plane. And so that's why sometimes when people look at this, I'll have, myself included, will hand draw what the cluster should look like in 2D and then realize, oh, it's actually the 3rd or the 4th or the 5th dimension. In total, we got 15 clusters across all of these 35,000 nuclei and 6 samples. And one of the first things I've kind of learned to do with these is just kind of look at what samples did they come from. And when I did that for this experiment, it was something quite striking. We, through a number of ways, we're able to look at CNVs and gene expression programs. I'll talk about this a little bit later on to say that these internal clusters were all the non-tumor cells. I didn't do any enrichment for any sort of tumor versus non-tumor because these are nuclei. I don't have cell surface to sort on. But the thing that really jumped out at me is if I color the cells by the mutation status of the original tumor, there is a very stark separation. Essentially, any dot or cell that came from a BRAF wild type tumor, I have colored here in red. any dot or cell that came from a BRAF mutant tumor I've colored here in blue. And again, I think you can see from across the room, there seems to be a real separation of these. Even in the non-tumor cells, and I'll talk about this in a moment, but you can see that this non-tumor populations here seem to be mostly red or mostly blue. And I think that has potentially important implications for what types of therapies might work for these patients. So the take-home point from here is we got tons and tons of nuclei across 6 samples, and they seem to separate out based on BRAF mutation status, consistent with the prognostic significance of BRAF mutation. Oh, too many. Got impatient. All right. So the next thing we want to look at, and something we'll commonly do with this type of analysis, look at cell cycle progression. The idea here, very simplistically being, and very beautifully shown again by Hugo, that cells that are proliferating might be bad actors. The common way to do this is pick a set of genes that are associated with G1 phase, G2M phase, or S phase, and score that as a big gene set, and then color each cell or dot by that score, and assign it a cell cycle phase. I hope what you can see here is that many of the cells in the top right are pink, which is the G1 or not actively dividing phase, and then many of the cells on the bottom left are in the G2M or S DNA synthesis phase, which are more actively dividing. I kind of let the cat out of the bag by advancing too aggressively, but what you can see here is the BRAF V600E mutant cells seem to have a significantly greater proportion of dividing cells. Maybe this explains, at least in part, why these tumors are more aggressive and outcomes are worse. And the other thing that I wanted to look at, given my interest both now in the past and in the future on RAS signaling and growth factor signaling, was some of the targets of that pathway, notably genes such as DUS4 and DUS6, which are targets of ERK directly, and genes such as ETV4 and SPRAT2, which are downstream of RAS more generally. What you can see is that these genes are in fact enriched in the BRAF V600E mutant cell populations. So again, this is a UMAP of this same set of cells. We're coding them or shading them based on the expression of the genes as indicated here. Anything in purple means that the cell has a high expression of that gene. Anything in gray means the cell has a low expression of genes. And you can see that the purple parts are all focused on the BRAF V600 mutant E clusters. So can't give a single cell talk without talking about immune system, even though I am very perhaps unabashedly not an immunologist, so I'm open to thoughts and feedback, especially with melanoma metastases. The immune system obviously matters a lot. And so we wanted to at least start to look at this in our data. And so I told you at the beginning that I figured out those middle cells were non-tumor cells. I didn't tell you how. One way we did it was with C and V's, copy number variants. We expect normal cells to not have those, expect tumor cells to have them. The other way to do it, and really actually the first beautiful paper from Ite Hiroshi looking at melanoma metastases, they actually use CD45 as a marker of the non-tumor cells. And so kind of piggybacking on that approach, if you look at CD45 expression in our clusters, you see that very nicely localized to this center set of clusters 8, 9, 12, 14, suggesting that those are indeed the microenvironment and not the tumor cells proper. And then if we look at some common markers of different T cell subtypes, here we have CD3 and CD4. You can see that the CD3 expressing cells are mainly up here, and the CD4 cells are both these clusters. Again, these are feature plots where purple cells have high expression of the gene, gray cells have low expression. So you can hopefully appreciate that. Probably have to be closer to the front of the room to see these, so I apologize. The other markers we looked at were CD8, which you can see expressed also in this subpopulation, CD163, which is a macrophage marker, expressed more so in cluster 8 over here. And then CD69, a marker of T cell activation, which is expressed specifically in this compartment here. I'm going to summarize this all in a moment, which is to say, going through all of this exercise, here's how we ended up assigning the cell identity of these microenvironment clusters. That cyan-looking cluster is CD163 positive macrophages. Cluster 9 are CD4 positive T cells. Cluster 12 are CD8 positive T cells. And then I didn't show the markers for cluster 14 because they're not immune cells, so I assume no one here cares about them. They're endothelium, which is PCAM and IGF BP7 positive subpopulation. And so the last data slide that I'll have for today, perhaps the obvious question is, how do these different subpopulations differ based on BRAF status? And it turns out there's something I think quite interesting that came out of this. I certainly didn't know this was going to be the story when I went through this data analysis, but I think it is going to become the story. If we look at cluster 8, which are those CD163 positive macrophage population, what I have here on the x-axis is the 3 tumors that are BRAF V630 mutant in blue, 3 tumors that are BRAF wild type in red. The y-axis is the percentage of cells in that cluster. The CD163 macrophage cluster 8, there are significantly more cells in this cluster from the BRAF V630 samples compared to the BRAF wild type samples. And we actually see somewhat interestingly the opposite in terms of CD4 positive T cells and CD8 positive T cells, where there are more cells from these lineages in the BRAF wild type tumors compared to the BRAF mutant tumors. N equals 6, N equals 2 in each subgroup. As a biologist, this is a smoking gun, but as a real scientist, I don't think this really tells us anything definitively other than this could be something to follow up on. So to conclude the talk today, I think at least 2 things I can say. Molecular status, namely BRAF and NRAS mutation, correlate with poor clinical outcomes in melanoma brain metastases. I think that BRAF finding, again, has been described before, the NRAS finding less so. Unfortunately, due to the heterogeneous nature of the therapy these folks got, it's really hard to correlate these mutations to response to systemic therapy. This is something I think we can do going forward. And then the other caveat here is I told you about this really great CBio portal way to access all UCSF 500 data and get de-identified information. Unfortunately, things like radiation, which is what I do, isn't in that system. So we're actually going back and now looking in detail at exactly what types of radiation these folks got, because I know about their surgery, I know about systemic therapy. Radiation is kind of the third part of that treatment paradigm. And then the second big picture conclusion is that at least in this small cohort of N equals 6, single nuclear sequencing suggests that there's differences between BRAF wild type and BRAF mutant brain metastases. And again, I think this can have important treatment implications for what types of immunotherapy and what types of targeted agents. So last slide, things we're working on right now. As part of our IRB and UCSF 500 data, we've also collected non-small cell lung cancer and breast cancer brain metastases with UCSF 500 data. So very similar idea, looking at how prognosis and potentially predictive responses to treatment might matter in those histologies. Again, I think it's really important to not put them all in one bucket as brain mets, but try to subdivide them out at least by histology. I envision a future where we do it by molecular status, but that's me being very starry-eyed as a junior faculty. The other thing we're doing is trying to integrate this with radiomic and histopathologic approaches. So I'm excited to see that Javier has autosegmentation up and running, because that will save us a lot of time in terms of extracting these features. But we do often end up in the situation where we don't surgically resect the brain metastasis and instead go straight up with radiation or with systemic therapy. I think it's a little bit of a cautionary tale in that there's now data coming out that up to 50% of brain mets might have a different mutation profile than the primary tumor. So using the primary tumor to guide intracranial therapy might not be optimal in up to half of cases. So it'd be great if our friends in radiology could help us to figure out what type of mutation this tumor might have. I think that could be really powerful. And then the last thing, and kind of a shout out to one of my fellow postdocs in the Abate lab, is doing spatial single cell analysis, a very obvious next step with all these single cell data. We have a workflow where we can actually get true single cell resolution in comparison to things that are out there right now, which are more 10 to 20 cell resolution. And we ran this on just a single brain metastasis from this cohort to give you a sense of what that data looks like. The other thing we did here was actually DNA mutation profiling rather than RNA sequencing. So we can look at V600E mutant clones. We all use RNA sequencing on the science side, but I know of 0 clinical decisions I make based on transcriptome signature. So I think DNA mutation really matters, and it's something we should look at directly. And so what I'm showing here, just as a kind of a shameless plug to Cyril and who's interested in collaborating with folks that are here today, is on the left side here, we have essentially the RET mutation. On the right side here, where the V600E mutation. What each spot here is an individual cell in space, and they're colored based on mutation status. And so this patient happened to have a synonymous RET mutation. And so that's in their germline and they're heterozygous for it. So all the green stuff is normal cells. The tumor in this scenario has lost that. So that's in blue. Those are wild type for that synonymous RET mutation. And then if we look and contrast that BRAF V600E, blue here is wild type. You can see that that blue wild type really maps spatially to that synonymous RET mutation. So those are the normal cells. And then everything in the other colors are BRAF V600E heterozygous or homozygous. Not a mutation we think of as being homozygous. In most patients, we think one copy of activated BRAF is sufficient. So I think these are the kinds of things the future of single cell can help us tease apart biologically in terms of heterogeneity in the lesion. With that, I will thank everyone and all the funding agencies that made this work possible. No man or woman is an island, and I am certainly no exception. So I want to thank everyone for their attention, and I'm happy to answer questions now or at the end.